Hey everybody, my name is Amir Mahari, and today I want to talk about my research project that I'm presenting at this conference. First, I want to give you a really quick introduction about the melanoma. Actually, melanoma is the deadliest form of skin cancer, and more than 50% of melanoma cancers have mutation in the BRAF genes. So in normal cells, EGF binds to its receptor on cell surface and it triggers a signal to the cell nucleus by the MAPK signaling pathway, and then cell start to divide, which is a normal process. But if we have mutation in MAPK signaling pathway, for example, here in our research, BRAF gene, it triggers signal to the cell to divide even if we don't have EGF binded to its receptor on the cell membrane. So for treating patients with BRAF mutant melanoma cancer, most of the doctors or physicians they prescribe BRAF inhibitor. We have different drugs, targeted therapy, different drugs for inhibiting the mutation. For example, in our research, our collaborators at Vandy University, they used vemurafenib drug as a BRAF inhibitor. When we use vemurafenib drug, when we add vemurafenib drug to our cells, initially in response to drug, cells start to die, which is a positive outcome, which is a really good outcome. But then the size of the tumor cells, this size of the tumor will increase. The size of the tumor gradually increase, then enter to the steady state level. We named it idling state. That's why we named it idle. It's because of that. Here in this steady state level, tumor cells, they are dividing and dying, but at the same rate. Because they are dividing and dying, eventually they get mutation, and at this time it's really hard to treat them. Here in this research what we are saying is that when tumors stop growing, it's the best time to attack. But in order to attack, we have to have detailed understanding what's going on with these cells. We have experimental result. We simulate our experimental result. Our simulation can successfully reproduce our experimental data. Uh, so here, after this point, we are going to use our model, our simulation as a virtual lab. And our goal for the next step, our next steps, for example, is to see instead of vemurafenib drug, what will happen if we use other drug? Or maybe we are going to test combination of drugs to see what will happen and test them in experiments to see if our model can reproduce or produce exact result as we expected or not.